Osmosis is a special type of diffusion. When you think osmosis, think water diffusion. Osmosis is the diffusion of water through a membrane. Still from high concentration to low, but it's a special type. We call it osmosis. And so water inside of our organs, inside of our cells, across any selectively permeable membrane, will develop a different pressure gradient, a different osmotic pressure, and wants to even out. Really, the salts and stuff do too. But if we think about a membrane like our cell, the water can passively diffuse through things. Big molecules, like an ion or something, can't. And so even though they both want to even out, that's just the nature of things. In these permeable membranes like our cell, only the water can fit through. So only the water can even out. It creates a pressure difference, a gradient difference. You ever eaten a lot of salty foods and then got really bloaty, or you just felt like you're full? It's because you are. It's because you differentiated that pressure difference, this osmotic pressure difference, and the water flooded into certain areas because they were full of those salts. There are special channels we have learned recently specifically for water. And so we thought water used to just go through the plasma membrane, and that was the only way it got into a cell. Now we know there are special channels called aquaporins. Aqua like water, pore like an opening, a hole. So aquaporin is the water pore. It's an integral channel protein that allows water to enter the cell when we need high concentration. Sometimes we just can't wait for that diffusion difference to occur. So because of this pressure difference, because of this difference inside the cell, we can begin to describe scenarios using these new adjectives. So we like to say, well, let's just say we have a beaker. I don't know if they can see that or not. No, I want to see me. Yeah, they can see that. If we had a cell in a, I don't know, dish of water, let's just picture one cell. I know it's kind of hard to imagine one cell here. These X's are going to represent a salt. I don't care what ion, let's just call it, just say a general salt concentration. The clear space is going to represent the water. Water inside the cell, water outside the cell. If our cell was in a solution that was much more water than salt, and we had much more salt than water inside the cell, there's now a pressure difference because they both want to even out. They both want to diffuse. But can the salts fit through this plasma membrane of the cell? No, they're too large. Water can diffuse though, so water is going to diffuse. It's going to balance out. And so in this situation, if we have more water outside than inside, and you think about diffusion and osmosis, move from an area of high concentration to low concentration, which way is the water going to flow, in or out of the cell? What do you think? In or out? Where is there more water? Outside the cell. There's less water inside. So it's going to flood the cell. Because those salts can't balance out. They can't fit through the plasma membrane. It wants to balance out. Now, if you, in this situation, the cell may burst because it gets to a point where it can't balance out. And so we begin to describe these solution types. And so solutions that have the same amount of salts inside the cell as outside, we call isotonic. Iso means same. Tonic is just in reference to the solution. And so isotonic, same inside, same outside. There's just a net movement. So we can imagine if this was isotonic, it had the same number of salts, the water movement would be equal. And so your blood cells are a little salty. 0.9%, about 1% sodium chloride. And so our blood has to remain a little bit salty, so these cells don't either dissolve or burst. A hypotonic solution is the first scenario where there's more water outside the cell than inside. Hypo means less. Hyper, if you think hyperactive, hyper means more. Hyper means there's more water inside of the cell than outside. Now, something that took me forever to understand with these adjectives, we're talking forever, because I didn't think about these for a long time, and you learn them in bio, and you don't use them for several years, and you hear them again. These words are just adjectives, like green, tall, short. Hypotonic, isotonic, hypertonic are just adjectives. So we could use it to describe the inside of the cell. We could say the inside of this cell is hypertonic to the outside. Or we could use it vice versa and say that it was hypotonic outside and hypertonic. And so when we're describing hypotonic, we just think of it as an adjective. But in this situation, we're describing the solutions. So in a hypotonic solution, there's less solute. So here's a picture. So in our normal salt, normal saline, if you've ever gone to the hospital, if you watched a hospital show and you're dehydrated or they're just keeping you full of fluids, they don't put water in your IV, right? They put normal saline. Or they have some fancy name, lactated ringers or something. But that is just 0.9% sodium chloride. It's to keep you normally hydrated. Now think about the water, or think about your blood vessels. And so in a hypotonic solution, if a cell's in a hypotonic solution, it's more dilute. So there's more water outside than inside. So the water's going to flood the cells. In a hypertonic solution, there's more concentration. It's saltier. Water's going to leave the cells and move toward that concentration because it says there's way too much salt. We need to equalize here. And so your water would flood the solution. So if you had low blood pressure, right, and they gave you a hypertonic solution, water's going to flood your veins and increase that blood pressure. Or some enemas are hypertonic. And so if you put a bunch of salts, or if you can't dissolve lactase, right, you can't dissolve lactose, with lactase. All those end up in the intestine. Bacteria have a heyday, going crazy with all that sugar, create a bunch of toxins, a bunch of gas, and there's a bunch of these sugars left out. You create a hypertonic environment. Well, your body is naturally, water floods your intestines, and then you experience the negative consequences. Maybe a little bit easier thing to think about. If you go to a beach, ever been to the beach before? The ocean water is really salty. If you spend a day in that water, your hair dries out, your skin dries out, because you've been inside of a hypertonic environment. All the water left your cells, all the water left and dried you out. And so we'll look at these again in lab, but it may help you to draw these a few times. Draw a cell and think about what it means to be more concentrated inside or outside. And think, well, water's going to equalize. Things want to move from high to low concentration, but the salts can't fit through the membrane. And so it's just a typical test question to say a cell is in a blah, blah, blah environment, a hypertonic environment. Which way does the water move? As an example, that's just a very stereotypical test question. And so you'll need to be familiar with these. We'll look at it in lab too. We'll do this again, and you'll get experience with it. But become familiar with these three. I did not. I don't know why in Gen Bio. I could never picture which one was which. But it's because I didn't realize these are just adjectives. So maybe that will help you. In a hypertonic solution, your cells are going to shrivel up. The water's going to leave the cells. They're going to shrivel if they're too much of a difference. And so when that happens in blood cells, we call it crenation. When it happens in plant cells, we call it plasmolysis or plasmolysis. Again, those are lab terms that will come up, so we'll see those again. Here's another